Good afternoon. I'm Leslie Ewan, an Associate Professor of Medicine at the Division of Nephrology at the Washington University School of Medicine, St. Louis, and a member of the AKINow Basic Science Work Group. Thank you for joining today's AKINow panel presented by ASN and the AKINow Basic Science Work Group. Today's attendees include nephrologists, fellows, nurses, administrators, and technicians from across the globe. During today's panel, Doctors Adriana Hung, Sanjay Jain, and Chirag Parikh will discuss how to build and best use kidney repositories. Here is our agenda. Following the panel discussion, I will lead a conversation based on questions which you, the audience, submitted during the registration process and through the Q&A option on your screen. So please feel free to add questions through this Q&A mechanism. Please note a few housekeeping items for today's panel. In acknowledgement of the changing nature of medical information and practice, please note this disclaimer. Here are the disclosures for all of the individuals participating in today's wonderful panel. All right, let's go ahead and get started. So biorepositories are very valuable resources for kidney research, but little attention has been paid to how to set them up. We are so lucky to be joined by a group of distinguished experts to join our panel today. Panelists, if you could please turn on your webcams. I'd like to introduce Dr. Adriana Hung, who is a professor of medicine in the Division of Nephrology and Hypertension at Vanderbilt University Medical Center. She's also the dialysis medical director of the Nashville VA. Dr. Hung's research centers on examining the role of genetic and environmental factors that contribute to CKD progression and outcomes in CKD. Dr. Veteran Program Publication and Presentation Committee and co-chair of the MVP Phenomic Working Group. Dr. Sanjay Jain is professor of medicine in the Division of Nephrology at Washington University School of Medicine in St. Louis. Dr. Jain's lab is focused on defining how the kidneys and lower urinary tract were organized at a cellular resolution to conduct their normal functions and the changes that occur in these tissues in disease states across the lifespan. By building upon his foundational knowledge and using as a benchmark to identify molecules that alter during aging and disease, his lab is hoping to glean insights into disease progression. He is the PI of the kidney atlasing efforts and the HubMap and KPMP consortia and leads the Pediatric Center of Excellence in Nephrology at the Washington University School of Medicine in St. Louis. And last but not least, Dr. Chirag Parikh is the director of the Division of Nephrology and a professor of medicine at Hopkins School of Medicine. He also directs the Precision Medicine Center of Excellence for Kidney Diseases at Johns Hopkins, and his research focuses on the translation and validation of novel biomarkers for the diagnosis and prognosis of AKI and CKD. He's the PI of Tribe AKD Research Network and has led several multi-center biomarker studies in adult and pediatric AKI. So let's go ahead and get started with our panel. And I'd like to begin with Dr. Parikh. If you could speak about your experience with Tribe AKI and the challenges that you experienced to set up this repository on your own, including issues like structure and process.
Thank you, Leslie, and thank you, ASN and AKINow committee to host this conference and inviting me to participate in this important topic. So this is a topic which is very close to my heart. If you are a translational researcher or enjoying the journey of precision medicine, the samples and the integrity of samples are important part of your research infrastructure. And my journey started in 2005. I finished my PhD in translational research and clinical investigation with Bob Schreier in Colorado in 2005. And the first R01 was developing novel urinary biomarkers for AKI. We wanted to replace serum creatinine, and the answer was in the urine. So the first few months, I called around a lot of biobanks, and nobody was storing urine. Everybody stores blood and plasma. And this is 20 years ago. And again, still urine is not very fashionable sample to store. So did not have much choice in a good way that I said I had to start my biorepository. There is no ready-made primary study where I can do it. And then the second question was, should I buy into somebody else's biorepository or start my own? And there are larger groups in cardiovascular or cancer groups within the institution who have a biorepository. But usually when you are going to be a PI, you want some flexibility, you want to change protocols, and that biorepository was built for some other purpose. So considering a lot of different things, I decided to start my own biorepository. And we started off with 2 freezers, and over the last 20 years, we have over 2.5 dozen freezers right now. And the journey is very interesting. It is not as complicated as it sounds. A lot of people want to go and say, oh, do you have some space? Can I do it? But if you're going to work with samples for many, many years, it's good to have some flexibility, some control, some customization that you can do with your science and your protocols, because your answers are in the sample. But if you're thinking about a biorepository, think about it as a biobank. It's a collection of specimens that is linked to some human data, some personal health information, and you are going to use it for health and medical research. And like a bank, you are going to deposit something in, which is your sample. So you need to know what you're going to bring in, the way you would put your assets or your money into a regular bank. And then the interest that you get is scientific. You'll get knowledge, you'll get new therapies, you'll get new tests, and occasionally you get a monetary reward, but most of the time the benefit and the dividends are scientific. And then you need the processes to get the assets out. So if you are thinking along those lines, you know you need a good structure where the bank is secure. You need processes to deposit and to remove it and to manage it, and then some QI. And I think Adriana and Sanjay will cover a lot of these different things. But if you are a single PI starting off and you want few freezers, all in terms of structure, you don't need much. You need like institutional support where they give you a lab space which has good electrical supply with a emergency backup, a good ventilation system, because they produce a lot of heat. So your integrity is maintained. And once you have that, the second piece is your informatics, your protocols that will decide how we are going to annotate the samples. And I think we'll cover that as part of this thing. And then some QI processes. So when I started first, it was hard. There was no examples out there. It was very early. But now I think the field is very rich with several biobanks, several protocols, and it's much, much easier to get it going. It was delightful in 2009 when Time magazine said that biobanks are one of the biggest investments that the community can make right now. They do this top 10 every year. Now the top 10 is all going to Mars or doing something with AI. In 2009, it was developing a biobank. And I think the community has seen a lot of benefit in various ways. So I'll stop here so we can do it. But the main consideration is build versus buy. And your scientific need and your journey should decide whether you want it for a short-term or a long-term purposes.
Thank you so much for starting things off. That was really great and touched on a number of topics that I think we'll delve into more. So next, I'm going to switch over to Dr. Jain. And Jeff, if you could pull up his slides as well, that would be great. And he'll touch into some issues related to how to set up the biobank, like with consent issues and quality control, things that will help make the biorepository flexible over time. Thank you, Dr. Jain.
Yeah. Thank you. Thank you for the organizers to giving us the opportunity to share our experiences. Thank you for also the very kind introduction and share some of the stories with what Dr. Parikh mentioned. I started our biorepository in WashU in nephrology division in 2007, and it really was when I was doing a lot of developmental biology and mouse studies and finding mutations that were causing developmental defects. And then I was looking to translate this to humans because we were finding that the developmental mutations were also making these animals susceptible to increased injury. But we were not able to find any samples. So we started as part of my recruitment, I started collecting samples from our local partnerships over here. And then we were part of the O'Brien Center. So I wanted to expand this in a more collaborative way. So then with some institutional support, we expanded this to institutional and then accessibility to worldwide. So what I would basically maybe show a few slides to summarize in general our experience. So to summarize our experiences over the years of how we're thinking with our involvement with not just only at local level, but as you heard, I'm part of the several consortia, including the Human Atlas project with the HubMap and KPMP for the kidney disease biopsies. So our center is also developing protocols that could be used for more new generation technologies. So in general, the questions we ask is when you're building a biorepository is like, who is it for? What is it going to serve? What purpose it is going to serve for? And how to make it happen, which includes questions of infrastructure that I think Chirag will also touch base about later on. And then do we need to think about applications not currently perceived? And this is a hard one. And this we have learned from experiences, both in the consenting process with patients, the usability of the biorepository, as well as new technologies are coming. And then how we sustain it. So the next slide basically touches on general points. So in purpose, for at least for a lot of our work or our users, we think about a lot of next generation technologies where we're looking for cell diversities, biomarkers, pathobiology, and validation. These are the kind of user requests usually get or people are trying to put in their grants asking questions. And then we think about sources. Where is the tissue going to come for the questions that people are asking? And there are several sources listed over here. And then really to think about trash in, trash out. And that has a whole background of things that you have to worry about when you are building this repository. And then factors range from accessibility, whether it's private or industry, that will determine the consent process. Patient burden is very important because if everybody's doing silo research, then we will have 10 people showing up into the patient pre-appointments or in the surgery suite trying to collect samples. And that is basically a big no-no for our IRB, as well as not good for our patients. So we have to think about ways that we can share and be efficient. Metadata, sampling, and quality control is very important. So this is a slide that we made for KPMP. It's about rigor and reproducibility. So you want to minimize variations. And this is where sample processing and preservation are key. And as far as patient metadata and phenotype also. And then tissue processing, which is the second circle over there, involves pathology processing and preservation. So you need partnerships, not only with surgeons, clinicians, also with pathologists and your local coordinator network. And these all impact the downstream analysis, which a lot of what we're doing now is for omic and molecular studies. Next slide. So this one basically tells you a little bit about some factors to consider in procurement, such as ischemia times. Preservation and processing includes what are the samples perfused, what media, fixative, freezing time, shipping, all these timestamps. Tissue morphology is very important, especially for molecular studies. What you are going to, how you are going to orient the specimens, what's the composition of specimens, and who is triaging it or QCing it. That can impact what your results are, what results you're expecting. And molecular is what is going to be used for. Do you want these samples for biomarkers, for RNA studies, for proteins, metabolites? That impacts what infrastructure you need in place. Next slide. And this is just an example of you need, when you're collecting these tissues, how do you know they're good? So you need some sort of a metrics over time to assess its quality. So this is just some of the omic studies we have done on the tissue collected. And these are Levy-Jennings plots that we make in KPMP. And you have to be within 2 standard deviations of the mean to assess how your sample processing as well as your assays are working. So this is one of the QC metrics we employ, and have some reference samples bank that we can over time use again to just benchmark our sample collections as well. Thank you.
Thank you so much for that. And one quick question before we move on to Dr. Hung. You had mentioned that the consent form, there's some considerations to think about when you're setting that up that could help with the flexibility of your biorepository down the road. And I think you mentioned industry and sort of the ability to use this for industry is something that you might need to consider for the consent form. I wondered if you could expand on that or talk about any other considerations that you were really glad you put in the consent or that you wish you had put in the consent when you were starting off.
Yeah, thank you. So over the years, as we evolve our science, the IRB board's policies need to evolve as well. So what we have learned is that our consent, first of all, most of our 90+% of our samples and clinical data are from consented patients. Over time, that for publications, as well as for applying technology, sharing data broadly, that the consenting patients was the best option. Otherwise, you will end up having done a lot of work and not really be able to share those results. And this also is important because when you are collecting or doing your studies, you cannot perceive all what metadata you need from the EHR. So if your consent addresses that you would be indefinitely can access patients' records for future, then you can go back and you say, okay, well, I was studying diabetes, but I wish I had some parameter for lung function, or anxiety meds, So those kind of things, right? So you want that in your consent broadly to be accessible, not only access retrospectively and prospectively EHR records, but also samples. That means a patient comes 5 years from now and you have lost to follow-up, but if you see that they have had other procedures done, then you can still get your the samples from them without bothering them again for consenting. So those open, and then there's increasingly partnerships with the industry and scientists, and so for those, if there's not explicit language that you can share the samples with industry, at least our IRB would not allow us to do that. So we have to go back, amend to explicitly say industry, and from then that point of time, then we can share samples with the industry. And then for costs, you have to tell patients whether there'll be any direct benefit to them or not. And mostly there's no direct financial benefit to them. So that's usually stated in the consent. And then the other issue is return of results. And that's a tricky one, like what are you returning? And because if you want to return them genetic results, then you also want to provide support for genetic counselor. So those kind of, for example, when we were doing pediatric genetic mutation studies, we explicitly would state that if we find a mutation, or in any patient, if we do genome sequencing, we find an oncogenic mutation that was not really intent of a study, but because we are surveying it and it has actionable consequences, then we would contact IRB or figure out a way to contact that patient. Otherwise, patients for research studies are not given results back.
Thank you so much for that. That's very helpful. And again, just to let the audience know, Dr. Jain has shared a lot of his references that include guidelines for things like tissue prep and processing that will be available to the audience early next week on the website. So now we'll turn to Dr. Hung, who has a leadership role and has been very involved in the VA's Million Veterans Program. So she's going to give a little bit of an overview of that. And Alicia, if you could pull up her slides, that would be fantastic. And then talk a little about involvement in national repository versus, building your own repository and how they can differ in terms of the questions you ask and the phenotyping.
Thank you so much, Leslie, and thank you so much for the invitation to join this panel. I think Chirag and Sanjay have done a phenomenal work in covering many aspects of what you face when you're building your own repository. And it's very interesting because as I hear them, I think you experience many of the same issues in a mega biobank with like MVP. And yes, they made them, and actually MVP and BioView made mistakes in their consents, and then many years later, you pay the consequences of the consent not being just right. And so it's so incredible. I could spend many, I will say, hours talking about on the different aspects and how it had affected sometimes the evolution of the study. But for the Million Veteran Program, let me tell you that this is an initiative of the Department of Veterans Affairs to advance genomic and precision medicine. They started enrolling in 2011, and they enroll across the country for I think it's about 80 VAs that have participated. They reached more than 1 million participants around Veterans Day. That's November of last year. However, they continue to enroll to see if they can enroll more females and underrepresented minorities. If you are familiar with the VA, you know that females are about like 7%. But one of the greatest things of this biobank is diversity in genetics. This is something that was absolutely lacking worldwide. And in this very particular sample here, which is release 4, that's 658,000 participants, 19% happen to be of African ancestry. By the way, what you're seeing in the pie chart is not self-declared race, but it's genetically impaired ancestry. So that gave us the opportunity to do further research in APOL1 that actually turns out to be very highly relevant as the characterization of a protective variant in an APOL1 gene called N264K, which protects against kidney disease even if you have APOL1 G1 and G2, so became the gene proxy for APOL1 blockers. And if, as you can see, there's 61,000 Latinos. That's actually the largest sample for Latinos as well. And so just to tell you what's available there currently, right? So there is, as I mentioned, genetic data in 658,000 individuals. This is done in 2 types of array, just regular Affymetrix, and one that has been customized to cover specific variants for minorities. There is a 10,000 participants pilot in whole genome sequencing that is amazing because they went really to very rare variants, so it's 80 depth. I may share some of that at the ASN. And they have currently 42,000 individuals with methylation data. That's very important because, as you know, environmental factors change methylation and impact gene expression. So it'll be super cool to see what comes from that in analysis of gene-environment interactions. They're doing whole exosome sequencing, metabolomics, and proteomics, but those are really baby steps. So it will take a while to get those back. So they're not in the near future. But one of the greatest things for the Million Veteran Program is the phenotypic data. It's linked to the VA electronic medical record, which has a wealth of information, so it's very granular. And more than anything, because this is a closed system, you have people that are being followed even for 20 years. So I can say that for us nephrologists that we always want to see like 40% decline in GFR or ESRD, this become a phenomenal electronic health record. But one thing also that they did is that they went beyond the electronic health record and they completed health and lifestyle surveys at enrollment. This is offering an incredible amount of data, sleep patterns, social determinants of health, physical activity. There is a food frequency questionnaire embedded in 352,000 participants. It is an incredible amount of important information that we as nephrology for sure want to use to understand how they impact kidney disease progression. I have this point of view that it's not just gene, it has to be gene and environment. So it's the same topic all the time. And so they also bring National Death Index, CMS, and many databases. They have a tumor registry created by the VA. They have a CART, which is after cardiac catheterization. Many others, actually, data sets from other studies in the VA system have been incorporated. So that's make it even better. The downside of here is that all this data, whether it's the phenomic data or is the molecular data or the genetic data is being deposited in an environment called Genesis, and Genesis is behind a VA firewall. And in order for you to enter into Genesis, you do have to have a PIV card. So you have to be either a VA employee or a WC. Genesis space is, by the way, de-identified. The data is going through a honest broker. But there is one aspect that is great, is that the VA has created something called the Data Commons, that is a computing cloud. And in that computing cloud, the phenotype has been fully de-identified. Yeah, you cannot go down to the granularity you will get in the CDW, but it's not bad. And this particular data commons is being created for non-VA investigators. So hopefully it'll be open to non-VA investigators soon. I know there's already 12 funded projects inside the space, and they're seeing their performance to understand the limitation, how friendly it is to use, and really if the data was enough to complete the different hypotheses that were included in those 12 projects. So that's something to look for in the future. I do want to highlight another initiative from that started as a VA initiative, but as a MVP initiative, but now is all VA and maybe beyond the VA. And it's this centralized interactive phenomic resource. And what this resource does is that it has definitions in many different phenotypes in a very comprehensive way. People are even including their SQL codes, their R codes, everything. So you go there and you say, well, I don't know what you want to study nephrotic syndrome, and you will find everything that you can pull from there and copy and paste it into your work area and just change the code so that it will run in your area. So that resource, it also has like functional visualization and it has many different tools. It's very easy to find. Just write Cypher VA in Google and you will find it. That's the link, but just write Cypher VA and you'll see it. I also want to recommend you to go to the MVP page that's publicly available. That's mvp.va.gov. Next slide. And I just chose to share one of the areas of that website, which is geared to patients. And this is something super cool because when we started the MVP, we had paper signed consents. But you can imagine for 1 million people, that would require buildings and buildings and buildings. It was just crazy and almost very difficult to handle. So they changed to e-sign, but I love the online signature. It's really good. And the patient can schedule when he's going to come to give you the specimen. And so people just schedule themselves when they're coming to another doctor's visit. They can also even send the blood, but they're not really doing that frequency. You can send it by mail. It's something called the Tasso T, I think. It's a device that you can send home. People stick it to themselves and it draws an EDTA too. And the surveys, if anybody is really interested in seeing the surveys, they're also online. Next slide. I just wanted to say that there's another biobank at Vanderbilt called BioView that is also very good. The one aspect of BioView that's the best is that they conquer a mirror image of the electronic health record, which is called the synthetic derivative. And so that provides you the ability to construct your phenotypes in a very granular way. So for me, that's the strongest aspect of the BioView synthetic derivative. They are going to reach really soon 250,000 people with whole genome sequencing. So talking about when you want to nest and use other, do and build studies within a larger biobank is easier for me in BioView. It will be easier for you if you work with the biobank in your own institution. If I were to try that at the VA, it'll be hard. I promise I will, but it'll be really hard. So next
Thank you,
slide.
Adriana, so much. These are great, sort of a great overview. And I think the MVP program has a lot of potential as well as BioView for our audience. And what we may do is put some of these slides on the web too for people to refer to. I was wondering if we may bring back just to webcam and go more into some of the logistic issues that I think some of our audience members may want to look into in terms of tracking systems and infrastructure and the finance to support and maintain some of these biorepositories that are kind of the nuts and bolts and the nitty gritty of keeping this, of starting it and keeping it going. And Chirag, I thought I'd turn over to you and see about tracking systems and what works for you, what doesn't, what advice do you have for our audience members who are thinking maybe starting my own biorepository is something I should do?
Absolutely, and thank you, Leslie. So once you have secured your physical space for the samples, reliable infrastructure, the next thing or equally important thing is your informatics that go around it. And once again, if you're thinking like a bank, you need to know every transaction, what's coming in, the details, etc. So I think about informatics in few ways. One is inventory. At any given point of time, you need to know where your sample is. There needs to be a good GPS around it. So which box, which shelf, which freezer, which floor, which protocol, etc. can be found out. It has to be linked to the study in such a way that when you are doing secondary studies, you can easily identify cases and controls or persons with certain outcomes, etc. There are also quality processes that need to be part of the informatics, and they include things where you can capture bias and chance. So, for example, if you are doing a study on biomarker and you say, I left the sample in the freezer at -80 till it was analyzed, and it could be in for 2 years, it could be in for 4 years, the sample could have shipped across the 5 participating sites. It could have been frozen and thawed few times, or you have used a daughter aliquot or a parent aliquot. So all these things need to be captured in your informatics system. So once again, when I was starting off, there were not many, and the FreezerWorks, which is out there, was not available. So we did our homegrown system with our programmer. I have a set of slides which give you the components of the infrastructure and the informatics. The software is also available for free download because we have published about it in the Journal of Informatics, but we give you the framework, and starting from there, now we have enhanced it quite a bit. So reach out to me if you need it, and now it's on SQL, and you can attach it to your hospital firewall, etc., or we can disseminate it through ASN, but it is available. There is a lot of joy in having your own system, because once again, the flexibility to what is there in your lab, the type of freezers you have, and the type of studies you are doing can be accommodated. A lot of times when you buy software from outside, not only are paying subscription costs per user, etc., but the flexibility goes away. So these are once again the pros and cons of it. But your informatics system should tell you about reports, locations, studies, protocols, should have good governance available. If a patient withdraws from the study, you know how to find the sample, annotate it, discard it, and also help you with the science that comes along with it. So these are the different considerations.
Thank you so much for those different things to consider, and then the pros and cons of sort of developing your, having that own software system for tracking. Dr. Jane, I'm wondering if you could also comment about the tracking system that you use and that you've developed, and has that changed over time as your biorepository has grown?
Yeah, thank you. Yeah, so what we do over here is we use a Washington University adopted specimen tracking system. It's called Open Specimen. And it's also used for a lot of cancer centers across the US and so forth. So it's a centralized system in the sense that we don't have to pay for it. So the university or the cancer center funds it, and it keeps it standardized for across all the studies at WashU. So it has all the features that what Chirag was talking about: specimen tracking, inventory, and so forth, and maps of freezers, so forth. So that's what we use for specimen tracking. It's over the years, it's evolved to become user-friendly for queries and generating reports, exporting files, and also adding your own forms to it. And then for clinical metadata, now we use REDCap, which is also centralized in some sense to collect metadata. The pain points over here that before REDCap, we were using a different system in a university, but then that was not sustainable. So then they migrated everything, and that was quite painful. And we're still feeling the pains of it because the structure changes. And so that's something to keep in mind. So that comes to sustainability. How are the things that you have are supported, because you want to, when you consent the patients and they say use for future studies and for understanding their disease, you want this outreach of what you're collecting for a long time. So you have to think about sustainability, how you're funding it, and those mechanisms to support it too.
Great. And I thought, Adriana, Dr. Hung, we could talk about, if you want to talk about, I know you have your own biorepository that you're developing, and any challenges related to either the tracking system or storage, and how you've sort of navigated those challenges.
Yeah, so I have been less innovative in the part of the tracking and what I use originally Freezer Works, and now I use Freezer Pro. So it works very similar to what Chirag is describing. One of the reasons why I chose to use that is because I can also involve other centers and we can share what we're inputting. I'm not sure if Chirag's, your system can allow that too. There you go. So it seems like Chirag's covers all the aspects of those systems. In regards to phenotype, I love REDCap. REDCaps give me everything. But when we're using really large data sets, sometimes I just switch to text files, and then I import them into statistical softwares. And so I keep it in a server, usually like an SQL server. But REDCap is where we handle everything. Mm-hmm.
Fantastic. And I think, the one comment that Dr. Parikh made about how he started off with, what was it, 1 or 2 freezers, and then expanded to now to dozens of freezers. And those freezers are not cheap, I'm sure. And so I think one of the questions that a lot of people may have is the financing of something like this, and what are the finances required to start and get something off the ground? And then what about maintaining and growing? Where does the cost come in? And how do you involve maybe some other stakeholders, the institution support, other CTSAs, where do they come involved to help maintain and grow this biorepository, which is really a great resource for the larger research community? Who would like to start off with that one?
I can tell you, a lot of times you don't have the full map. You can see that the next few steps, and you don't see the whole staircase. So when we started, I did have institutional support where I got a lab space and had an NIH grant that covered the cost of the freezer and personnel. And then the key is to right size it for the type of work you are doing, the type of science you are doing, and the number of people who are going to use it. So I can tell you my story, but again, it's a bespoke that I don't know whether it is reproducible, because the science was broad. We did not know we will get the answer in our first round, the 2 or 3 IL-18 or KIM1 or NGAL biomarkers we're looking for. We did not know whether there would be answers. We knew it may be a 10, 20-year journey. So we were thinking of increasing capacity all the time. And at some level, if your science is good, the success comes in where people are ready to invest in your biorepository. And what by that I mean is you get grants from NIH, you get ancillary studies where others are interested and are willing to do cost recovery for you in terms of accessing those samples. American Heart Association, a lot of mentees write their grants, the K Awards, dozens which have gone out, they all come back and feed into the system. it does happen with the institutional backbone where they allow you to, you don't have to worry about electrical and ventilation and other things. If you can maintain the personnel and the samples, I think the biorepository can go. And so if you're doing a registry, you may have a different consideration. If you're collecting samples as part of a clinical trial, you may have different considerations, and you have to make sure that your finances meet your science. Leslie, you are muted.
Sorry, you touched on a number of important things related to personnel that's required to help maintain this. But first, want to go back over to Drs. Hung and Jane and see if either of you have anything to add about supporting financially a biorepository. I mean, with the MVP, I know that's not. But you've started your own, Dr. Hung and Dr. Jane. You've really grown yours, and how that. Did you start with institutional support, and do you have to have some reinfusion of that throughout the way as you grow?
Yeah, so I think one of the important thing is that you have to be willing to invest your own, like somebody has to champion the repository, right? So you can't, it's a big undertaking. Financial investment is there. We started with O'Brien Center to grow it, and then when the O'Brien Center phased out, then our division committed to some funds to sustain the effort, and also supplemented by some of our own grants. And then also because we did service for the investigators, so we also asked in some cases investigators to put in some effort of some of the personnel if they were substantially involved in their grants, and if they got the grants and they had effort. And then to that, we have also established a cost recovery mechanism, which follows the guidelines of the university and the NIH to recover costs of services that at subsidized cost that we cannot supplement, because it is an expensive undertaking.
Great, thank you. Dr. Hunt, anything to add? And then I'll also throw out, there's a question that says, for those of us who are on K grants and the cost of a brand new freezer is prohibitive, is it suboptimal to buy refurbished freezers? And I think that might be, that makes me a little nervous to think about a refurbished freezer, but maybe an option. But Dr. Hung, anything you want to add with the costs?
No, I started with funded grants, and I did get a lot of support from the CTSA. So that's always a good source. And then, as Chirag was saying, then other mentees that get their own K23, and then they can help.
Yeah, I can come in. I wear my administrator hat right now because end up doing. So for a freezer, it's usually over 5,000. So the NIH doesn't charge indirects on it. So whenever you get a chance to buy a freezer on your grants and you have accounted for it, that indirects will become part of your direct. So it's almost like getting 40% off on your grants. But still, the life of a K awardee is always challenging. Like Leslie said, rather than buying a refurbished freezer that you'll manage it and come in middle of the night and pay people to do call and for maintenance of freezer, might be good to have 2 or 3 K awardees come together and buy a freezer, or make a plan with your mentor that can I have the space now, and as I'm getting independent, I will become I will have my own setup like we do with our children who are going into becoming independent. So I think there are various ways, but rather than going into a refurbished freezer, think about other innovative ways where people will help you. If you ask, people will help most of the times for a K awardee.
Great, thank you for that. I think we had, there was one other question that we may get to towards the end about sort of the success of biorepository, but I wanted to continue along the lines of sort of the nuts and bolts, and there's sort of a cost recovery program that I think a few of you have referenced. And, you know, is it cost neutral? There was a question in the audience about whether it could be for profit. And I don't know if, my guess is that very little in research is for profit. But maybe you can charge, say, industry different rates specimens than academics, and how do you deal with that issue?
Yeah, so sorry, should I or go ahead?
Go ahead, Dr. Jane.
Yeah, so we have all these different models. So for industry, we have a different rate, and for collaborators and junior investigators, we have different rates. And sometimes for like small pilot studies, especially for junior investigators and so forth, one of the center what does actually give them also at no cost, you know. So that's basically the structure. But you can never be according to NIH guidelines and university guidelines, it's not for profit. So you cannot over the years show like you have made a profit. So because otherwise you have to adjust your rates. So yeah.
Great. And if we can move on, there's another question from our former AKINow leader, Dr. Samir Parikh. Pathways are available to young investigators who want to generate repository-based preliminary data for grant applications?
I can go in. Sure. And Samir and I have worked ancillary study together, so I think it's a lead question for me. Anyway, I think if people get a chance to work off a existing biobank, especially for junior people, it's a delight, because the time is the most important thing. Even if you start your biorepository today, it can take several years to get the outcome. So let's say you are doing a CKD study and you want patients ending up on dialysis transplant. You're talking about 3 to 5 year median follow-up. So even if you put your hard work in today, you don't get the rewards right away. Forget all the other sweat and other equity that you'll bring into the table. So if most of the NIH studies, the PIs are willing to share the samples for the right studies. So if your question fits into existing studies, you should definitely reach out to the PI through the NIDDK project officer or the PI directly, who's the corresponding author, and we are delighted to help. So as Samir knows, we have this Tribe AKI Consortium, Assess AKI Consortium, KPMP. We probably helped over 100 investigators, and it's very fulfilling, because a lot of times new knowledge emerges, new careers get developed, and new blind spots in our knowledge that cleared up, which really serves the reason you started the biorepository. So I'm a big fan of ancillary studies. NIDDK actually had a program for 10 years where they were giving out R01s and R21s, where you are using up the samples. And you should go for it. New technology on old samples is a very good way to succeed.
Thank you very much for that. And this actually feeds really nicely into some, I think, information that Dr. Hung has about examples where junior investigators have been able to leverage these biorepositories to really move their science and research forward. Dr. Hung, would you like to comment more? I think you're on mute now.
Okay, perfect. Thank you. Well, there's different ways, and we try to mix them and merge them together. So, for example, having my own biorepository, right? We have very deep phenotyping in insulin, glucose clamps, and we have samples of every type, and very, very specific phenotypes. So sometimes you can talk to a biorepository, and if you want to do extra things, let's say urine proteomics, right? So you can say, I'm going to do urine proteomics and plasma proteomics, and I'm going to do genetics on my guys, right? And so with this information, I am going to try to apply my proteomics approaches to your biobank. So that is easy, right? It's just that when you're writing your grant, you can show the strength. We're going into deep phenotyping. You can really answer mechanistic questions very well. The other way around is using big data approaches, right? the big data approaches are hard, but are really available to juniors. You can write phenomenal papers. I love, for example, the druggable genome. Jefferson Triozzi had a beautiful paper in JAMA Network Open on thiazides, answering the question that he didn't agree with the fact that thiazides were not useful for kidney stones. So you can do a mix and match. You can put an AIM1 and AIM2 that go really to the biorepository, and then use your own biorepository to address, saying, I'm using this to address this question in a deeper level. And the other way around is that if you are in contact with the person that's building their biorepository, let's say BioView, we can go and say, well, I'm going to take a random sample of the biorepository and I'm going to do urine proteomics. And they will say, well, that's fantastic, right? You will still not violate the identify aspect of the biobank. You will add to the biobank and you will develop expertise in a new omics. So that's the third way you can go.
Fantastic. And I think that kind of gets into this question, this use of, your own biorepository versus utilizing a national one. And it looks like Dr. Hung has been able to successfully mentor a junior faculty into embedding your own and kind of using that with a national. I think it is worthwhile maybe chatting about some of the pros and cons of utilizing a national biorepository versus starting your own. And Dr. Jane, I know that you had talked a little bit about sort of the advantages of using the institutional biorepository. Maybe you can chat about that in a bit, and then we can have Dr. Parikh talk about the advantages of making your own, which you sort of alluded to earlier. Dr. Jane?
Oh, sorry, I thought I was going after Dr. Parikh. Yeah.
I'm sorry.
Yeah. So,
Yeah.
So for us, we started as a private in a biorepository, but we grew into an institutional and a national or international accessible repository. The advantage of those are, first of all, is that the outreach of the patient samples becomes much broader because we do collect different kinds of samples and different kinds of kidney disease, not just focus on one kidney disease, because our user base is more broad on kidney diseases. So that's why, so that was the advantage of institutional in the sense that it is supported by the institution, but it's locally controlled. So our systems are centrally sort of made. So for example, our track is an institution system, but it's locally managed. So we have full control of our systems and our repository, because the advantage of that is, as Dr. Parikh was saying, because you have immediate access and you have liberty to access your repository at will. Because if it is a third-party source, then there are hurdles that can delay your access or storage and things like that. So it is institutionally, as far as infrastructure is concerned, but with with a local liberty to control it.
Great, thank you. And Dr. Parikh, if you want to sort of speak about the advantages of making your own biorepository, and do those outweigh the hassles of not only the funding, but also the personnel, and what personnel that you need to sort of maintain that and manage your biorepository?
Yes, once again, there are pros and cons of having both your homegrown versus working with a national biorepository. The clear advantages of a homegrown are the efficiency and the flexibility of the sample collections and protocols that you can have. So, for example, we have had at least a dozen times where a basic science investigator comes in and says, I found this good protein in the setting of AKI or CKD or GN. Do you have a set of samples where we can see whether I should put my postdoc on it? Because if it is not in humans, it's probably not worthwhile. We'll keep searching. And some of these have been very successful collaborations into careers and grants and papers, and like YKL40 is a classic example that that has emerged from such a collaboration, and there are numerous like this. The second benefit is pandemics and other COVID, where having your protocols and biorepository, you can pivot very quickly to collect samples, expand your protocols, and try to find knowledge around these new emerging diseases. In terms of personnel, it is like a bank. You need investigators who are benefiting from the biorepository, who are meeting regularly to make sure that the health and the biorepository is good, where you need to update it in certain ways. We need to change freezers or informatics, create scientific protocols and committees where we are sharing the samples efficiently, updating the informatics, etc. You need personnel who will help remove the samples, thaw the samples, create daughter aliquots, ship the samples, etc. So, and plus you need a disaster recovery plan. So I would say at least twice a year, one freezer, even if it is new, will break down suddenly at the wrong time. It will be the weekend, and your on-call staff, including the faculty, have to come in and have a plan in place so that you empty that freezer before it has become too late. So we have audit systems in place where those could be done. So you need personnel for these components. But if your ancillary study mechanism is very busy, you need more personnel. But if it is not busy, and you are getting 1 or 2 studies per year, you don't need that many personnel to remove the sample. So it kind of has to fit the demand.
Thank you so much. And Dr. Hung, would you like to comment on your thoughts in terms of the balance of this?
Yeah, I just want to add that your own small biorepository versus a national biorepository, they have different goals, I will say, right? I think that when you're doing big data approaches, you are more in the discovery side, right? So you're going to detect signals, you're going to learn about many pathways that need to be further investigated. So whatever you learn, wherever you discover, in many occasions, you have to bring it to a small repository or to a basic scientist, right? Because the functional aspect of what you saw needs to be proven. And so I think it's different. I love big data. I think it's incredible. I get all excited when I get these findings that make sense. And I sometimes you will have never gotten there if you were hypothesis driven. So one is very discovery type, and one is very specific to a set of hypotheses and to understand mechanisms. I think they're complementary.
Fantastic. And it looks like we're wrapping up, I think, for this session. And I do want to, first of all, just really thank our panel of distinguished guests, Dr. Hung, Dr. Parikh, and Dr. Jain, all of whom have successfully navigated this realm of creating biorepositories. I do want to let the audience know that a lot of their references and slides will be posted on ASN website at the beginning of next week, and you should be getting an email about when that should be live. So again, thank you very much to the audience, to ASN, and to our wonderful panelists for a great discussion.